The cell cycle begins with interphase. It is the longest phase of the cycle. It consists of three steps: G1, when the cell prepares to replicate the DNA, S, when the DNA is replicated, and G2, when the cell prepares for division. Importantly, during the S phase, each chromosome is replicated and paired with an identical sister chromatid.
Mitosis. The first stage of mitosis is prophase. During prophase, the loose bundle of unwound DNA and protein in the nucleus, called the chromatin, condenses down into thick bundles called chromosomes. At this point, the nuclear membrane is also breaking down. Outside of the nucleus, centrioles align at opposite ends of the cell and begin to secrete long protein strands called microtubules. The next step is metaphase. The nuclear membrane has completely dissolved by this point, and the chromosomes are in the cytoplasm. They line up across the middle of the cell in a line called the metaphase plate. There, they are met by microtubules from the centrioles. Some of these microtubules attach themselves to the chromosomes. These are called kinetochore microtubules. Others stretch past the chromosomes to the other end of the cell. These are called non-kinetochore microtubules. In anaphase, the shortest phase of mitosis, the microtubules retreat towards the centrioles, pulling the sister chromatids apart. Since each double-stranded chromosome contains two copies of the same DNA, the end result is that there are now two identical sets of genetic information on either end of the cell. As this is happening, the non-kinetochore microtubules expand, forcing the centrioles further apart, which causes the cell to stretch out. In telophase, the last stage of mitosis, the microtubules continue to push the cell apart. Meanwhile, the chromatids gather in opposite ends of the cell. Two new nuclear envelopes form around them, and they unwind back into chromatin. This is the end of mitosis, but not the end of cell division. Cytokinesis is a separate stage from telophase, although they occur at the same time. In cytokinesis, the middle of the cell begins to contract, forming a cleavage furrow. The cell pinches itself off and automatically reseals its membrane, forming two new cells that are both genetically identical to the original. Both cells are diploid, meaning that they have two N chromosomes, the correct number for life. They are now ready to divide themselves if necessary. Mitosis is important because cells must divide in order for organisms to grow and sustain themselves. Since DNA carries instructions for all of an organism's life processes, it is important that all cells receive the same genetic information.
Meiosis differs from mitosis in several ways. It produces 4 haploid daughter cells, in comparison to mitosis's 2 diploid daughter cells. It also has one more stage of division than mitosis. In meiosis 1, homologs pair and divide.
And in meiosis 2, the individual chromosomes split into their separate sister chromatids. Also, meiosis is unique due to the genetic diversity introduced by the crossing over of chromosomes at chiasmata during prophase 1.
Meiosis. Meiosis ensures the diploid number stays constant generation after generation when gametes meet and fuse. Also, it introduces genetic variations and thus increases diversity, adaptability, and evolutionary potential. Meiosis differs from mitosis in both form and function. Meiosis reduces the chromosome number by half in order for the organism to produce haploid gametes. Unlike mitosis, it has 2 series of divisions, meiosis 1 and meiosis 2. Chromosomes condense into a more dense form, the nucleus begins to dissolve and synapsis occurs, leading to the formation of chiasmata along non-sister chromatids. This allows the exchange of genetic information, also known as recombination or crossing over. The nucleus finishes dissolving and kinetochore microtubules attach to the centromeres and move the chromosomes to the middle of the cell along the metaphase plate. Chromosomes move towards opposite poles of the cell on the kinetochore microtubules, which shorten. Each chromosome still contains 2 chromatids. Microtubules dissolve and the chromosomes have reached opposite poles in the cell and revert to chromatin in the reformed nuclei. Cytokinesis occurs, producing 2 daughter cells. The stages of meiosis 2 are similar to mitosis or meiosis 1, except for the fact that sister chromatids which previously comprised each chromosome are separated into different cells, thus producing a total of 4 daughter cells from meiosis.